Hello, everyone. My name's Tom. I prefer Tom to Thomas, but you can call me Thomas if you'd like. The introduction's already been made on my behalf, but I work for a fund called Maki, and we invest across Europe. And whilst we are generalist as a fund, I think within the team we have areas of interest that we've ended up kind of specializing in. And for me, one of those areas ended up being biotech. For the simple reason that it's really interesting. And a lot of the things that people work on in the biotech space, I find just extremely fascinating. I mean, you're really at the forefront of, the absolute cutting edge of what's happening in science, what's happening in some of the most important fields that exist. So, I'm very privileged today to be moderating this panel. I will probably do my best to speak as little as possible, because the last thing that you want to hear from is a VC. I'm very well aware of that. But I am very privileged to also be able to welcome to the stage today two people who will know a lot more than I do about this really interesting topic of what's happening in biotech. And so I, without further ado, would love to welcome to the stage both Juozas Nainys and Rolando Delgado. So please, can we give them a big round of applause? Welcome to the stage. I'm glad that I now have someone else here. Feels a lot better now. So look, guys, this is trying to answer what's happening in a field as big as biotech in half an hour is quite a difficult challenge that we've set ourselves. And I think before we jump in and talk about some of the really interesting innovations that are happening, I think one of the things that's quite unique about biotech, or the field in general, is that there's a long way to go from an initial idea or from some initial early research through to being able to call something commercially viable. And in a way that's not true, for example, if you're talking about, I don't know, an HR tech platform, for example. And I think that it's one of the things that kind of often people sort of overlook about the space, particularly from an investor's perspective. I think it's easy to kind of sometimes forget that something that might be quite far along in, for example, clinical trials is still quite a long way away from having like a real-world impact. And I thought maybe we could just start by, for anyone in the room that's not a biotech expert like myself, maybe talking a little bit about what that process looks like, what that journey looks like from, really like that early stage through to commercialization or commercially viable products. So Rolando, maybe we can start with you. Would love to, it's always difficult because there's never one route, and it's not as simple as saying this is how it works in every case. But I'd be curious, to maybe share with people a little bit more about what that process looks like, how we go from, step 1 to step 100, as it were.
Right. Yeah, thank you very much. Just in a few words, yes, my name is Rolando. I'm representing MGI Latvia, which is a company operating in the field of genetic sequencing as, so to say, producer and developer of genetic sequencing equipment. So that's kind of the reason why we more or less know something about biotech, so to say. But biotech as a field is extremely interesting how it has grown so fast, because, for example, in 2010, if we compare the number of companies listed in the NASDAQ index, so to say, since 2010 until now, this number has doubled, even maybe more. So it's an extremely booming industry. I would also say that COVID as a pandemic has also kick-started that extremely fast, because when a pandemic is happening, who tries to save the situation, who tries to save the world? It's biotech. Yeah, in any kinds of ways possible, whether it's vaccines, whether it's more efficient testing for the disease, or in other interesting fields like for example, telemedicine, contract tracing, and of course pharma, and etc. Yeah, so indeed, biotech has been overlooked by investors for some time because of the long development stage from idea to product, especially if we're talking about, let's say, vaccines. I think most of us are now much more educated about vaccines than we were 2 years ago, and we know that it takes some time, so to say.
Yeah, by the way, I've done a terrible job. My only job is to introduce the two individuals, and I didn't actually do that properly, so apologies, Rolando. That was terrible from me. But yeah, I mean, you're absolutely right. I mean, how many people knew about incubation periods and about all these kinds of things, 2 years ago? And in some ways, it's really benefited from that limelight. So Juozas, I'll do a slightly better job this time if I can. So you're obviously the CEO of Droplet Genomics. You can maybe tell a little bit about what you guys do and your angle in terms of how you kind of came to biotech.
Yeah, I guess we're a more kind of traditional biotech startup in the sense that we're commercializing a new technology, liquid handling technology, without kind of expanding too much. We're trying to make kind of high-throughput exploration of biology, single-cell stuff easier. I know that sounds complex, but our path was very typical, right? We built on years of research experience that was funded by grant funding, and it's, few inventions are really serendipitous. I think it really just, knowledge needs to accumulate. And I'll tell you a little secret, when you read the published paper, probably they got it to work once and published, and when you need to make a business and a product out of it, a lot of money and a lot of time needs to go into it. And this is, kind of what you mentioned, VCs often don't necessarily understand unless they're specialized VCs. But, be it technology development or clinical trials, it'll take time. And when you read that somebody, hey, discovered something new in cancer and we're going to cure all cancers in the next 2 years, that's not true. So I think, the cycle is long, that needs to be appreciated, but the potential is also huge. The things we'll be able to do in 20, 50 years, it's fascinating.
It is indeed, and we'll absolutely get to that. I think that kind of the progress that's been made, even in the last decade, is absolutely staggering. And when you read a little bit into it and you spend a bit of time, like myself, coming from, not coming from a biotech background, it is extremely exciting. And also, unfortunately, feels quite important and pressing, because there are some issues where we really do need to try and make progress, and so it feels like the time is very much ticking. But so why don't we start then with regards to answering the question. I realize that we've not actually got to the question in giant letters on the screen yet. So what is happening in biotech? Well, why don't we start with that very, very early stage. So when we're still in the kind of early stages of research, we're still quite a long way away from anything materializing in a commercial sense, but that there's really exciting and promising developments. I mean, there's so many that we could talk about. I mean, we obviously briefly had a chat about this before, and the danger is that there's kind of too many things. But I would love to maybe come to you, Rolando, and I'd be curious to hear if there's one particular thing, in the last 6 months or so that you've seen that's popped up, which you think has got real potential, and kind of what also is that kind of real-world application that might result from this kind of early research that's happening?
Yeah, I mean, from some, so to say, some recent research, I was a couple of days ago actually reading an interesting article also on The Economist and just researching a bit more about this topic casually, so to say. But what came up there is that, interestingly, it was called like the fountain of youth is being researched now. So precision medicine is something which is not new anymore, but it's still exciting field and very booming. But there has been dedicated lots of research towards methods of how we can prolong people's life, also quality of life, of course. But there were various interesting things like through lymph nodes, stem cells, for example, opportunities how to repair damaged internal organs without any particular invasive procedures. So yeah, that seems something interesting that has been coming up in the recent time. So we'll be living longer, hopefully healthier.
Yeah, it's interesting. I mean, epigenetics is another one of those fields which, when you read a little bit about it, is really exciting. It's staggering, the idea that you're actually able to make your cells younger. I'm probably slightly butchering the way of phrasing that. That's probably, if I've offended anyone, I apologize. But in very sort of layman terms, being able to actually reduce the age of your cells by kind of enacting certain daily routines or improving your health, etc. Something that you brought up there, which I think is really important to touch on, is you mentioned that it's no longer a particularly early or young field at the moment. Is it fair to say that in general, biotech, it's not quite as simple as calling it kind of linear time progression? Because actually really what happens is, you might have a piece of research that is in its early stages. It might then take 10 years before there's another breakthrough. And then on top of that, someone might discover something new, which means you have to go back and slightly readjust. And Juozas, I'd be curious to maybe just come to you on this and hear your thoughts on, in the work that you're doing, for example, with Droplet, how much of a linear journey has it been? Or is it simpler to say that it's kind of, it's a bit more choppy?
So it is actually quite a bit choppy, as you say. And probably, maybe 2 minutes on a single cell story. So single cell field started 13 years ago. And it was, there were 3 people in the world doing it for like 10 years. Because there was no technology that could deliver a good solution. That technology came around. It turned out to be droplets. I know all about it, but I'll not go into deep. But that technology came around. The field, it boomed. And probably none of you are aware of it. There is a project as large as the human genome sequencing project going on. It's called the Human Cell Atlas, where people are sequencing every single cell in a human body, right? To create like a proper atlas and to see how are we different between one another.
Wow.
And this was only possible, as you say, because something happened. A few fields collided, technology, kind of molecular biology came all together, and it enabled this. Now this will take another 10 years to complete, but once we will know what the reference human kind of cell atlas looks like, what a disease atlas looks like, we'll be able to much better understand cancer and any other disease actually on a single cell level, which is a fundamental level of human biology.
Yeah, I mean, you said it absolutely right. And I'm also be curious to hear from you, Juozas, if there is, at the moment, any particular piece of research, anything in the early stages that you've been tracking, you've been watching, perhaps something that you've read that you think's got real potential that you're kind of very excited about.
So I think there is a real, so I'm mostly watching kind of technology progress, right? So not necessarily what people discover, say, in immunology or in cancer biology. But I think spatial kind of analysis is coming into focus a lot. And spatial is where you say, well, you take a slice of tissue and you can pinpoint a particular region in the cell where a particular mutation is. And I think that's really going to be powerful in diagnostic context in a probably near future, like 5 years or so.
And I guess an important question with something like this, particularly for those of us who are not necessarily that familiar with the industry, how does that information kind of pass through between different people who are working on this research? So you might have someone, for example, here in Riga, someone in London, someone in New York, someone in Tokyo, or teams in those different places who are working on, the same research. Is there collaboration? How much of it is kind of people working almost against one another? In your experience, what have you found is typical in that regard, Rolando?
Yes, our company has been involved also in a lot of collaborative projects, actually a lot of European funded projects as well. Our role there is to be the industry partner who helps to provide the technology and the capacities, because a lots of scientific partners and public partners, they have of course capacities for sequencing, for analysis, automation to some extent. But we, as a technology producer, of course, our capacities are top-notch through the roof in that sense. So that's why we try to make it available, and we also have been involved in various projects where we help to handle large amounts of samples. And this is the part which you mentioned, where we need to exchange this information with various collaborators and key opinion leaders within the project. And of course, practically, usually, if we were to do some kind of sequencing on our platform or so, then in that case, of course, this data is encrypted. It's anonymized. It's not. We do not get this kind of data, and we do not intend to do that as well. This is the project leaders do that. We just provide the technology to be able to do it. So of course there are some security restrictions, data regulations and all of that, which everybody needs to comply with.
Yeah. No, I think you're absolutely right. Okay, so why don't we then turn attention towards the slightly later, again, I know it's not, I'm sort of already destroying my own point about this kind of linear progression, but talking about slightly later progress or things that are closer to kind of commercial realization, as it were. And Juozas, maybe we can start with you on this one. I'd be curious to hear if there's anything that you think is on the cusp, as it were, of being ready to kind of enter the public sphere, and you know, whether or not there's anything that you, like any thoughts you have on that, or kind of whether you know there's still some progress that needs to be made.
Yeah, so probably two things. You know, obviously vaccines is a big thing, and everybody is focusing on how mRNA vaccines will change cancer treatment. But I think I actually have a lot of potential in other fields. And for example, HIV vaccine, they just started the trials, mRNA vaccines for HIV. And you know, that vaccine has eluded people for 40 years. So if we're able to make a breakthrough there, I think it's going to be pretty powerful. And another thing that was in the news just recently, over the past 3 months or so, is the kind of transplanted pig heart into humans. That went successfully, well, to some extent, but I think it was really the first proof of concept that showed that we should properly pursue this. So I think, again, in a few years, you know, transplantation is a massive challenge. There's, you know, literally not enough by orders of magnitude kind of available transplants. So I think that's also something very important that's happening.
Yeah, well, what was it in the end? How long did the chap survive for? It was something like 3 or 4 months, was it?
But they're not yet disclosing the reason why he died, so it's, you know, maybe it was not the heart, who knows, right?
Yes.
Hopefully.
But no, you're right. I mean, the idea that you would be able to take the hearts of animals that we are able to produce at, you know, mass scale compared with humans, and then implant them is, I mean, it's, it sort of sounds like science fiction.
But it's here today.
But it is here. I know, it's extraordinary. So Rolando, what about you? You know, I don't know if CRISPR and gene editing is a topic that you would consider to be kind of in the later stages, in the early stages still. But yeah, I don't know if you have any thoughts on CRISPR, or if there's anything else perhaps that you know recently have been looking into on the more commercial side of things.
Ooh, if we talk about CRISPR, I think that's a rabbit hole, so to say, because even though the technology is not very recent, it still has a long way to go, and it has a huge potential. But you also mentioned this, that in sequencing and in genetics and related fields, many things have been done already a long time ago. But to some point, we reach a point where the technology becomes available, and then suddenly there's an explosion and we can do a lot more. Because previously we just couldn't do it due to lack of technology or so. And I think the same with CRISPR. I mean, if we are to cure genetic diseases, it's difficult to imagine curing genetic diseases without some kind of genetic modifications. So it opens a lot of opportunity. But then also it's always about the second discussion, what about using it for the proper measures, and when it may become like a dangerous tool, or so to say. But I mean, it's a question of regulation, because if we look at it from that perspective, I was actually discussing this also a bit with a colleague, and we came up with an interesting comparison. I mean, electricity, discovered a long time ago. It's dangerous, right? It can be used to power an electric chair, or it can be used to power a house. It's all about regulation, and I think it's the same with gene editing.
No, I think you're right, because obviously you can't have a conversation about biotech or about anything similar to biotech without talking about those ethical issues, and it's always something that pops up. I wonder if I mean, Juozas, to what degree do you think, you know, in this case, like that you've personally, for example, or Rolando, actually, it kind of goes to both of you, have been involved with trying to help, you know, governments to understand some of that regulatory, you know, work? Because it is difficult to unpack, particularly for people who are often creating the regulation, who are not, you know, not scientists by trade. And you know, what work is done behind the scenes? You know, what more can be done as well to try and improve that kind of cross communication?
I think there was quite a bit about that on the last panel, right? How when we're talking about data from our side, we just try to tell them not to be afraid of new technologies, because yes, you can use it for good and bad purposes, but without new technology, there is no progress. So we need to kind of constantly reevaluate it. And I think that's probably one of the main bottlenecks in biotech, is the regulation and that transition to, today we can, it is possible to choose the eyes of a baby, it is possible to cure genetic defects, but how ethical is it to do that, and what should be the safeguards? Nobody knows, right? So that is holding us back, but probably should be holding us back. So I guess these type of conversations is what we try to start. And
yeah, well, I mean, look, that dovetails quite nicely, actually, onto another topic, which I think is really important, when discussing a lot of the exciting new innovations and some of the new commercial work that's happening, is also just to stop and talk a little bit about those bottlenecks and about what at the moment, are those bottlenecks. What are the things that are, quote unquote, kind of hampering progress across the biotech field? And this is open to both of you, but maybe we can just start with you, Rolando. Would be curious to hear what your thoughts are at the moment in terms of what some of those bottlenecks might be, or what some of those things that are slowing progress on a slightly wider scale.
Yeah. I mean, well, since both me and Juozas are working in the sphere of genetic sequencing, I could just elaborate that one of the main bottlenecks there is, of course, data processing, because creating a lot of data, sequencing a lot of data is no longer a problem. In particular, our company, we specialize in developing sequencing instruments for high capacities, high throughputs, and high amounts. So it's no longer the problem to create enormous amounts of data. Like in the previous panel, I heard it was said that more data you have, the cooler you are, right? But that's no longer the problem. The problem is, what do you do with this data? Because you need to interpret it, you need to put it to some kind of use, and if you want to use it more efficiently, then you also need a lot of reference data, because to find what has changed, what has happened within the genome, you need some kind of reference to compare it to. There are lots of exciting, interesting startups who are working to deduce this problem, but still it's limited to the amount of references we have. So it comes to the point that we still need to do lots of sequencing and population genetics projects.
And Juozas, what about you? What, like, for you at the moment, outside of that kind of data processing point, which I think is absolutely valid, what are some of the kind of bigger bottlenecks that you see more widely kind of across biotech?
I think in general it's the fact that biotech is built on kind of old generation technology, and I'll give an example. So immunotherapy, amazing, right? But the challenge there is pure logistics. So you have to take cell sample from a patient, you have to do something to the cells, you have to put it back into the patient. All of this has to be done under sterile environment. There's special medias that need to be produced somewhere. Everything needs to be done in kind of big flasks. There's people involved. There's clean suits involved, clean rooms. It's just, how do you deal with that, Even with these kind of very advanced therapies, they still need to rely on this fundamental kind of infrastructure, and there has been little innovation in infrastructure, to be honest, lately. So I think that is a bottleneck, but people are working on it, so probably there will be some innovation in that space, and again, in the next 5-10 years.
Yeah. And something that I think one of you mentioned, I can't actually remember which one it was now, is on that data, on the data processing point, I would be curious, and again, this is open to either of you, be curious to hear if there are any kind of horizontal innovations, as it were, that have happened in the last few years. It doesn't need to necessarily be that recently, that have really advanced progress. So, for example, some of the machine learning implications that in terms of being able to process the data and being able to understand some of the information that we're receiving. I think that's one kind of obvious one that jumps to my mind. I don't know if either of you have any thoughts on that, about some of those more horizontal innovations that have been going on.
I think one kind of innovation is, and it'll start not from data, but it'll circle back to data, is the fact that just data acquisition is getting cheaper and cheaper, and you know, it beats Moore's law by magnitudes, right? If we talk about sequencing, and what that means actually is that you can construct better databases. You can sequence more people, you have more reference, and that allows you to understand, you know, what one tiny mutation means in the context of population. You know, when we talk about human genome, it's just so much data there, and we don't know, you know, 10% of what it means, really. So, you know, AI will not help us much there, because you still need to feed something into
exactly,
AI.
you still need the inputs.
I think, yeah, it's a very big challenge.
Yeah, I mean, we just need a lot of reference data, because we've also had some of our collaborators working on these solutions by using AI, of course, and of course it's possible to create AI algorithms that scan through quickly thousands of reference data, and they can just check and match it with the particular sample, but still that is limited to the amount of reference data that we have. If there is no reference data, it will not find anything.
Yeah. No, I think it's a really interesting point. I think it's very easy to forget that it's not magic. Artificial intelligence.
Unfortunately, no, yeah. So one interesting, actually, way, probably this is a linear progress in a way, is people are using AI to look into literature, right? Because nobody can read millions of papers, but AI can. So if you give him, hey, I found this mutation, and you know, please read all the literature that exists and tell me what you find, AI actually tends to find pretty interesting stuff, because it's from different fields, and you know, once you see the 5 relevant articles, you can make that connection, you know, do the human intellect thing and connect the dots.
Yeah, and I think, you know, going back to that point around collaboration, particularly kind of when there's, you know, big language differences, you know, we talk, of course, most people here, I assume, are from somewhere around Europe, but when you start to get to Asia, for example, and you have totally different languages with even different alphabets, etc., and being able to, you know, scan a lot of that information rapidly and you know, perhaps find some information where people are working on something in Japan or people are working on something in Korea, and being able to like put the dots together, I think that's really interesting. All right, well, we are running out of time, so why don't we end on, I guess, a nice positive note, and talk a little bit. I mentioned we would come back to it and prove myself right. Talk a little bit about progress and kind of what, you know, how much progress has been made in the last decade. You know, maybe we can start with you, Juozas. What is something that you feel, you know, in the last, let's say, decade or 15 years has really taken a massive step forward? And you know, yeah, well, we can just start with that.
Yeah, I think, you know, I started with that. That's the shift to single cell resolution. Now we look at every biological system, at least in the human context, at a single cell resolution, and we can make completely different kind of discoveries than we ever kind of thought. You know, every system we look at, we find new cells, we find new drug targets, and I think that's super powerful. And that is roughly the last 10 years or so. Okay,
so single cell is Juozas, and what about you, Rolando? What is that? What's, you know, maybe talk a little bit about progress, whether it's in your field of genomics or something else. Would love to hear your thoughts.
Well, I mean, overall, I think it's exciting, the progress of precision medicine. Of course, genomics is a big part of that, but precision medicine overall, let's say, the genetic sequencing technology price is decreasing very, very rapidly. So it means that we can very soon reach the, like to say, €100 per sample, $100 per sample or so, and at that point, it can become part of a public health service like screening, just like your typical MRI, CT scan or something like that, just another analysis available there. And of course, there are lots of synergies there, government, insurance, and all of that, but of course, there's a big number of ethical and discussions and all of that. But the positive point there is that we will be very, very soon able to live a life with a very high quality life, be very healthy, and prevent any problems before they have started. Catch them and catch them early.
It is the holy grail, and you know, I know that for some people in the audience, at least for myself included, you know, there is a question. I think people sometimes question if it's even possible and whether or not we're ever about to get there. But I think, look, thank you both so much for your thoughts. It's been, I mean, it's such an interesting topic. I could talk about this for so long. There is so much you can talk about, and obviously we've had to be really high level with a lot of the discussion. But hopefully, at least people can walk away knowing that we are nearly at the point where we will be living forever. So if you take that one thing away from you this week, hopefully that's the thing. So thank you both. Rolando, thank you so much. Juozas, thank you so much. And yeah, enjoy the rest of the event. Thank you, guys.